All right. Hi, everybody. I had to make sure I was recording. Um, this is your TA review for exam 2 that is coming up in just a few days. So we'll just start off with a cute picture of my dogs because of course we would. This is, Barley, he studied a lot. He's really into it. He's excited to take the exam, but Honey still has quite a few questions. So, um, let's get into it. This is a lot of material to cover, so just a fair warning, I have 92 slides. Hopefully I'll be able to go through this kind of quickly. Again, this is just supposed to be a review, but again, I always want to point out that this may not be fully comprehensive just based on your class section, but I do hope that it helps just get general concepts across. So first, we're going to start off with chapter 2, and this is going to be a continuation of aqueous chemistry. I thought this was really cute. So it's, hey, oil, do you want to hang out? Sorry, I can't mix with you guys. Which leads us into hydrophobic versus hydrophilic. And so hydrophobic and hydrophilic are opposites, and the word stem hydra comes from the Greek hydor, which means water, and therefore hydrophobic materials are water-fearing, and they don't mix with water, but hydrophilic means that they are water-loving. So if we think about it and we put oil in water, we've all done this, we have a separation, and that's due to the fact that water is polar. So if we put a non-polar substance like oil into it, then there's going to be a decrease in entropy, or more organization is going to occur. And this is going to create kind of a cage around the non-polar substance. And this is basically going to lock the water molecule into hydrogen bond arrangements around the substance, and that's what keeps it from mixing. So again, think about this as organized hydrogen bonds of water around the substance. So when multiple hydrophobic molecules are being added to reduce the amount of entropy that is decreasing, these nonpolar molecules will actually be lumped together. And this clumping or aggregation is called the hydrophobic effect. So instead of having multiple of these, air quote cages around all of them, the hydrophobic effect is going to be considered a weak force that maintains 3D structures of biomolecules, but all of those molecules are going to kind of clump together, and there's only going to be one cage around them. Whenever we think about polar molecules, those are going to be able to form hydrogen bonds with water and then be dissolved versus making aggregates like you see here. So one thing we can kind of quickly talk about, and you guys know we will go through lipids in a lot of detail here in a little bit, but most lipids are amphiphilic, and they can form things called micelles, which are super cool. And so an amphiphilic or an amphipathic, that means that a molecule has both hydrophobic and hydrophilic portions to them. So here you can see this molecule that has a long hydrocarbon that is nonpolar or hydrophobic. And then the polar group here has a carbon and 2 oxygens, and that's what makes it polar. And so if you have several of these molecules with a polar head group and a nonpolar tail, then you can form something called a micelle. And so the polar head group is going to face the aqueous solution and form hydrogen bonds. And then the nonpolar hydrophobic tails are going to turn in and create what we call a micelle. And I don't know if you're like me, I'm sitting here in my robe with no makeup. And what I used to take my makeup off was something called micellar water. And this uses this technology to remove dirt and makeup from your face, which is super cool, and just a really neat application of this chemistry. And so next we're going to move into acid and base chemistry. And so the first thing is that when we think of pure water, some of that water is actually going to fall apart or dissociate into a hydrogen ion, and then into a hydroxide ion. And for example, here, if we have two water molecules, then one may dissociate, and the hydrogen, it doesn't typically exist by itself, so it will go and associate with another molecule, and that's going to create something called a hydronium ion, and then you will have your hydroxide ion. And so when water comes apart, we have about 10 to the negative 7th hydrogen ions that have disassociated and 10 to the negative 7th hydroxide ions. So if you remember this from Gen Chem, the brackets will actually mean concentrations. And so we can describe this by using a constant called the ionization constant or the dissociation constant. And we can see that the dissociation of water is 10 to the negative 14 total, with 10 to the negative 7, again, being those hydrogen ions, and 10 to the negative 7 being the hydroxide ions. And this kind of thought process and constant, remember it's a constant, it's going to be the same. This is where the pH scale actually comes from. And it's looking at the amount of hydrogen ions compared to hydroxides. And so the pH scale, and that's annoying that that's in the way, but the pH scale is a logarithmic scale. And so pH is the negative log of the hydrogen ion concentration. So when we have a high concentration of hydrogen ions, the pH is going to be lower. And then when you're at a pH of 14, you're going to have a lower concentration of hydrogen ions. So the higher the pH, the lower amount of hydrogen ions. The lower the pH, the more hydrogen ions you're going to have. Another kind of important thing to remember is that the scale is logarithmic, so very small changes in actual value. So, 3 to 4, that's a 10-fold difference. So changes in this pH scale can be quite dramatic, again, because it's on that logarithmic scale. So hopefully this is familiar to you as well. So I'll be quick with it. I don't know if any of you guys work in labs and have to pH something. It can kind of be a really annoying process, I have to admit. But if we want to increase or decrease the pH of a solution, we can do that. And it's pretty easy. It's just sometimes you overshoot them and it's a mess. But we can either add more hydrogen ions in the form of an acid, or we can add more hydroxide ions in the form of a base. And so, again, you can adjust the pH by adding these acids and bases. And in biochem, we define an acid as a substance that can donate a proton, and a base is going to be a substance that can accept a proton. And so in biochem, we think of weak acids like acetic acid that's going to dissociate to a small degree and only release a few hydrogen ions. So it only releases a few hydrogen ions. It doesn't dissociate too much. So we're going to call that a weak acid. So to determine how weak or how strong an acid is, we need to think about its tendency to ionize or its tendency to dissociate and lose that hydrogen ion. So you can have a K, remember we just talked about dissociation constants, so you can have a K for any acid in general, and you can look at the concentration of the base and the concentration of the acid and essentially determine the pH at which you have equal concentrations of both. So equal acid, equal base. These values are going to be constants. And so in order to figure out what's called the pK, you will take the negative log of that constant, and that's going to give you the pK value. So this example here is showing you acetic acid. And fun fact, acetic acid is I'm actually quite fond of because my dad works at a chemical plant. He's in the business side now, but he used to work as an engineer down in the plant, and he would always come home smelling like acetic acid. And so I actually am really comforted by that smell, which if you've ever smelled it, it is not pleasant. But anyways, so you'll get a pK value, and that is specific. The pK value is going to be a specific pH where you're going to have equal concentrations of the acid and the conjugate base. So you can see that worked out here where you have the K, you take the negative log of the K constant, and that is going to give you the pK or the pKa. You may see it written as pKa. And again, what the pK is, that is going to be the pH where you have equal concentrations of acid and equal concentrations of conjugate base. Unfortunately, I am a lame and I don't have the cool technology to write out equations for pK and pH. But my advice for you is to practice, practice, practice. You guys have a ton of problems from class and homework, and I made it through undergrad based on YouTube videos. So there's a ton of like Khan Academy videos out there that can help walk you through these calculations. Maybe one day I'll be cool enough to get an iPad with an Apple Pen to be able to write and talk through things, but today is not that day. So this is just an important note. So the pH of a solution is related to the pK. So in a solution of an acid with a low pK value, so that's going to be a strong acid, most of the molecules are in the unprotonated form. So strong acids will dissociate and lose their protons easily. That's the basic of what we're trying to get at with that top sentence. Strong acids easily dissociate and lose their protons. So for an acid with a high pK value, which means that it is a weak acid, most of the molecules will remain protonated. So this means that in order to pull off protons, you have to be above that pK value, which again, if it's really high, you're not going to see that quite as often. So this is the Henderson-Hasselbalch equation, and this is just showing that the pH is directly related to pK. So when the pH of a solution of an acid is equal to the pK of that acid, it's the relationship between the pH and the ratio of acid to base. So again, I'm lame and don't have the technology to write out these problems, but please go over your practice problems and the posted lectures. That can help a lot with this as well. I just think verbally walking you through it's going to be extremely confusing. So watching the lecture again and seeing it be worked out would be extremely beneficial for the Henderson-Hasselbalch equation. So when we talk about buffers, buffers are really cool. And so they have the ability to resist changes in pH within a system. And so weak acids actually make great buffers. And remember, pK is when you have equal concentrations of acids and of base, so equal numbers of acids and bases. So if I add a lot of a strong acid like hydrochloric acid, in a situation where we're sitting at the pK, then there's not going to be a drastic change in pH. So the best place for a buffer and the most efficient for buffering capacity is going to be at that pK, because again, you're not going to get a drastic change in pH. So the buffering capacity is considered to be 1 pH unit above and 1 pH unit below the pK. So if we think about, the acetic acid, which again, thinking about my dad, thinking about acetic acid, the pK is 4.76. And so the buffering range is going to be 3.76 to 5.76. So next we're going to move into protein structures. Proteins are molecular tools, and I actually study proteins in my PhD research. So if you ever want to talk about like how we research proteins, I would love to talk to you about that, because that's what I've done for 6 years, and what they're doing within a cell. So proteins are super, super cool. You can have a ton of different functions within different types of proteins. So you can have enzymes. So these can have catalytic function. That's what I think the protein I'm studying is. We think it's actually an enzyme. And you can also have things like structural and membrane functions. Another project of mine is actually studying some membrane proteins within a specific organelle and how they help to get molecules into that organelle. And so proteins can be distinguished by the number and composition and sequence of their amino acid residues, which we talked about in for the last exam. And so what's really important here is that if there are mutations in those amino acids, there can be a loss of protein function. And going into this, you guys should know, hopefully you guys already have them memorized, but you do have to memorize the amino acids. So I'm just going to run through them pretty quickly and point out some highlights. Again, there are a ton of fantastic tools online for learning how to memorize these. If you don't have access or haven't been able to find those, reach out to me or to your lecture professor, and we can kind of point you in the right way. There's a ton of like flashcard softwares and that sort of thing. All right, so moving into amino acids, proteins are polymers of amino acids. So amino acids have, again, that central alpha carbon that we've studied so much. And there's going to be a hydrogen in one position and a carboxylic acid that is deprotonated or carboxylate at a pH of 7. pH of 7 is physiological. And so if the pH, for example here, if the pH is 7 and the pK is 4, then you would show if the pH is less than that of the pK, then you're going to have the protonated form. But if the pH is greater than the pK, then we're going to have the deprotonated form at a neutral pH. And so that is why here we are showing the deprotonated form. Again, that's because if the pH is 7, the pK is about 4, then the pH is greater than the pK. So again, deprotonated form. So we typically draw the amino acids at a neutral pH. And another thing to point out is that we also have another weak acid as part of the structure, which is called the amino group. In the example, this is protonated. The pK must be greater than its pH, and the pK of this is 10. So if we're sitting at a pH of 7, that's going to be greater, so it's going to be protonated. So we can actually have chirality. So the amino acids are asymmetric. So we can have chirality for 19 of the amino acids and the 4 groups around the alpha carbon. So we can think about handedness. So we have right-handed and left-handed forms of the molecule. We don't typically think about the rotation in this. Again, as biochemists, we try to think of things as really simple and not worry about it. So the easiest way to think about this is for the L enantiomer, you're always going to have the amine group on the left side of the molecule. So L for left. And I just think of amine as starting with an A. And so that's going to be the first thing. So L enantiomer is going to have the amine group to the left. The D enantiomer is going to have the amino group to the right. Just a fun fact, L is the most common in nature. And this has to do with the machinery of translation. And a lot of times in bacteria, we will actually see the D enantiomer, and that may give them some sort of advantage in the host to have the other isomer. So when we're thinking about classifying the amino acids, we can classify them in a couple of different ways. So you can have nonpolar, which contain hydrocarbon groups with no charge. You can have polar, which contain functional groups that are easily interacting with water through hydrogen bonding. You can have polar acidic, which have side chains and a carboxylate group that are negatively charged at physiological pH, again, 7. And you can also have polar basic, which is going to have a positive charge at physiological pH. So just to run through these again, you guys hopefully by this point already have these memorized or are hardcore working on that. So these are the nonpolar amino acids. And so amino acids with hydrophobic R groups or nonpolar amino acids tend to occur in the core or the center of proteins, and that's because they're hydrophobic. We're going to start with alanine. So alanine has just a simple methyl group with a side chain. Its 3-letter is ALA, ALA, and its 1 letter is A. And it's one of the most abundant amino acids because it's so small. Valine is next, and it is a branched chain amino acid. Valine, its 3-letter code is VAL, VAL, and the single letter is V. It has 3 methyl groups on the side chain. Phenylalanine is the next one we're going to talk about, and it is actually an aromatic ring. And that aromatic ring actually comes off of the methyl group. Its 3-letter code is Phe, PHE, and then the single letter code is F. So, just think about the sound spelling of that, phenyl, F. And this one, just kind of a note, is that it's really bulky in nature. Next we have tryptophan. And so tryptophan also has an aromatic ring, but it contains a nitrogen. And so the overall structure of this side group does have hydrophobic character. The 3-letter code is going to be TRP, so trip, and the single letter code is W. So one of those weird ones that doesn't make a whole lot of sense. Leucine is the next one, and this is also considered a branched chain amino acid. Its 3-letter code is LEU, and its single letter code is L. Isoleucine, again, is a branched chain amino acid. Its 3-letter code is ILE, and its single letter is I. The next one we're going to talk about is methionine. And so methionine is really interesting because it actually contains a sulfur in the chain. And though the sulfur has an unshared electron pair, the overall structure of the side group does have hydrophobic character. Its 3-letter code is MET, M-E-T, and its single letter code is an M. Proline is not actually an aromatic ring, but it connects back to the amine group. So that's really interesting. It looks like it may be an aromatic ring, but it's not. Its 3-letter code is pro, and its single letter is P. And what's interesting about this is proline is considered a helix breaker. So it actually does not play well in secondary structure, which is really interesting. And so next we're moving to the polar amino acids. So these are amino acids with polar R groups. We have serine, which has a hydroxyl group. Its 3-letter code is SER, and single letter code is S. Next we have threonine, THR, and T. Again, some of these you're just going to have to memorize. Tyrosine is another one that looks or that has an aromatic ring. And you can kind of think of this because if you kind of flip it around, it looks like a tire swing. So tyrosine looks like a tire swing. And its 3-letter code is TYR, or tire, and the single letter code is Y. And again, I can remember why because the shape of a Y looks like a tire swing to me. So, you've got the 2 ropes hanging from the tree, and then down is going to be where your tire swing is. So I just think about that. Asparagine is going to have a carbonyl and an amine, nitrogen, and its 3-letter code is ASN, and its single letter code code is in. Next, we have glutamine, which has a carbonyl and an amine nitrogen. And the 3-letter code is going to be GLN, and its single letter code is another one of those weird ones, and it's Q. So glutamine is Q. So amino acids with polar R groups, and these, under certain conditions and the presence of nearby groups, can actually ionize and become charged. So cysteine is CYS or C. This can participate in disulfide bonds or covalent bonds, and those will only happen between two cysteine molecules. So histidine is another one of these, and histidine, its 3-letter code is H, IS, and its single letter code is H. The pK value of histidine is actually 6, which is close to neutral, and it acts as an excellent acid and base, so it can donate or accept protons. And kind of a good way to remember histidine is that the imidazole ring, so that ring coming off of it has 2 nitrogens. And that's kind of how you can distinguish it, is it has 2 nitrogens in the imidazole ring. So amino acids with charged or polar R groups, basic and positively charged are going to be lysine, which is LYS. And again, this is another weird one with the single letter, but it's going to be K. Arginine is the other basic positively charged group. Its 3-letter code is ARG, so pretty simple. But its single letter code, again, is going to be R. So you can just think about it when you're saying the name arginine, single letter code is R. It's a pirate's favorite amino acid. Acidic and negatively charged, so both have carboxylate groups, and these are going to be aspartate, or ASP, and then again, a weird one, this single letter code is D. And then we have glutamate, and its 3-letter code is GLU, and the single letter code is E. I just think of glutamate, it sounds like glue. So whenever you have your 3-letter code and your single letter code, it spells out glue. Finally, we have glycine. And so glycine has an R chain that consists only of a hydrogen atom. And sometimes it's classified as hydrophobic. However, it's neither truly hydrophobic nor charged. And it can also be grouped with the polar amino acids as well. So it's an interesting amino acid, again, because it has that R chain of only a hydrogen atom. So amino acids are linked via a condensation reaction during translation. And this happens through peptide bonds. The backbone, again, this is kind of the easiest way to remember it, is that you're just going to have repeats of nitrogen, carbon, carbon, nitrogen, carbon, carbon. The alpha carbon is going to be where that R group is located. And as we link amino acids, we now start to call them residues. So in a chain, each individual amino acid is going to be called a residue. We do lose the charge on the carbonyl with this peptide bond. And this is not spontaneous, and it actually occurs through a process through translation on the ribosome. Amino acids are, again, linked by peptide bonds to form a polypeptide. The N-terminus is going to have a free amino group, and that can be charged and carry a charge. And the C-terminus will have a carboxylate group, and again, that can be charged as well. So the charging of these groups will depend on pH and pK. As we start to think about naming these polypeptides, if you have 2 amino acids linked together, you have a dipeptide, 3 would be a tripeptide, 4 a tetrapeptide. And as we start to build up, so 8 to 20 amino acids, you have what's called an oligopeptide. And then when we start to get into our bigger proteins, you have above 20 amino acids, you would call this a polypeptide. And so remember that if you're looking at the N-terminus right here, what you're looking for is that NCC pattern to help you identify where each amino acid starts. So here we have our NCC, end of amino acid 1, NCC, beginning of amino acid 2, end of amino acid 2. And that pattern just continually repeats. NCC, end amino acid, NCC. All right. So you can also calculate charge of these peptides. And again, I really, really want to stress you guys should practice calculating charge. You can feel free to email me. I can try to direct you in the right way in terms of videos. We can meet, whatever you need. But I really recommend going back and looking at the lectures. You guys also had the in-class assignments where we walked through these kinds of problems. So I'm sorry I can't show these to you in the review, but just one thing that's important to remember is when pH is less than pK, it's going to be protonated, and when pH is greater than pK, it's going to be deprotonated. So that's just some important things to remember. I'm going to grab some water really quick. All right, so there are 4 levels of protein structure. So you have your primary structure, and so this is going to be the sequence of amino acid residues. You have your secondary structure, which is going to be your spatial arrangement of that polypeptide backbone. Then you can have your tertiary structure. So this is where you start to get into the 3D structure of an entire polypeptide, and you take into account those side chains. Next, you can have a quaternary structure. So not all proteins have quaternary structure, but this is the spatial arrangement of different polypeptide chains in a protein that has multiple subunits. So sometimes you'll hear about proteins having multiple subunits. Again, we'll kind of talk about that here in a minute. All right, so secondary structure. I thought this was actually really great. I just Googled secondary structure and found this sheet. So you can have an alpha helix or a beta sheets. So going through alpha helix, these are, again, in a helix shape. So they're right-handed coiled rod-like structures. They have hydrogen bonds that form within the polypeptide chain. The R groups of the amino acids are going to be oriented on the outside of the helix. And you can also have a single alpha helix, or you could have multiple. And there is only one type of alpha helix, which will make sense when we go into beta sheets. And certain amino acids do or are preferred in the making of alpha helices. And so beta sheets, kind of like the name suggests, they are sheet-like in structure. And the beta sheets are formed by linking 2 or more beta strands by hydrogen bonds. The R groups can be directed both inside and outside of the sheet. So they kind of flip-flop. And beta sheets cannot exist as a single beta strand. You must have multiple. And these beta sheets can be parallel and anti-parallel or mixed. And I will show you that in the next slide. And again, beta sheets also, similar to alpha helices, beta sheets do prefer specific amino acids. So beta sheets, again, like I just said, can be parallel or anti-parallel. So in anti-parallel, as the name suggests, you have beta sheets going in opposite directions. But in parallel, the beta sheets actually align into the same direction. So we can start talking about tertiary structure and protein stability. So protein folding is really important, and and can play a role in function of the protein. And this folding actually depends on non-covalent forces. So you got to think, if you have some hydrophobic residues that are in the unfolded protein, those, when it gets into an aqueous solution, are going to be folded up and protected by the rest of the protein. And so that's what gives you your tertiary structure. These can also aid in stabilizing or be stabilized by other factors that include ion pairs and also disulfide bonds. So these ion pairs and disulfide bonds can help in terms of tertiary structure. And then finally, you can have quaternary structure, which again, not all proteins have, but large proteins can actually be constructed in increments. And so I like to think of these as Lego blocks. You can have multiple Lego blocks that build one protein. And so this allows for some proteins to be assembled outside of the cell. Having a quaternary structure can help minimize the impact of errors in transcription and translation, because again, you have these multiple subunits that are acting together. And you can also have sort of a regulation effect. So the subunits can influence and regulate each other in what's called a cooperative behavior. And so this is just showing you some different proteins with quaternary structure. So you can have, 2 different subunits, 3 subunits, and so on and so forth. And so here you can see a monomer has no quaternary structure. You can have a dimer, so that's going to be 2 different subunits, a trimer for 3, tetramer for 4, again, so on and so forth. And so that brings us to protein structure. And so whenever we talk about protein structure and function, one thing that we can talk about is myoglobin and hemoglobin. And so myoglobin is a classic globular protein. And here you can see some different models of it showing its structure. And you can also see in purple, we have what's called the heme group. And the heme group is what allows myoglobin and hemoglobin to bind oxygen. So functional proteins that have a heme group, so that is where the oxygen is going to bind. Myoglobin is only made up of one peptide, so it's considered a monomer, but hemoglobin actually has multiple, it's a tetramer, and so it has 4 polypeptide strands. And they do have a different capacity to carry oxygen, which is really interesting. And so I just mentioned heme, but heme is a prosthetic group found in myoglobin and hemoglobin. And heme allows for myoglobin and hemoglobin to bind oxygen. And the main thing I want to point out here is this iron found within the center of heme. And so this iron, so myoglobin transports oxygen with that iron in the heme group. And so the oxygen binding depends on the oxygen concentration. So the more oxygen you have, the more oxygen is going to bind. And so here, this diagram is just showing you oxygen that is going to come in and bind to this heme group. And so myoglobin binds oxygen in a hyperbolic trend. And so the functional saturation we call Y, and that is going to be the proportion of myoglobin, the proportion of myoglobin molecules that have bound to oxygen. So bound over total. And so this gives us the equation of a hyperbolic curve. And so when we're talking about saturation, you have full saturation to nothing bound, and then you have your oxygen. As we increase the amount of oxygen, you can see that you end up having more oxygen bound or more saturation. And so myoglobin and hemoglobin are similar in their secondary and tertiary structure. And so this is just showing you the alpha subunit of hemoglobin, the beta subunit of hemoglobin, and myoglobin. Myoglobin can be seen in the green, and then alpha globin is in blue and beta globin is in red. And so you can see that their structure is very, very similar. However, they only have about 18% identity in their primary sequence. And so this is really interesting, especially to me, because I study a protein in a early branching eukaryotic pathogen where its sequence is unlike anything else that we've seen. However, when we start getting into these higher structures, we see that it has a very similar structure to another protein. And we think that's how it's functioning. So the sequence can be quite different, but you can still have similar secondary and tertiary structures. And so whenever we compare how oxygen binds between hemoglobin and myoglobin, you see, again, we have that hyperbolic curve when we look at myoglobin. However, whenever we look at hemoglobin, we see a sigmoid curve. And so kind of for comparison, myoglobin, its binding curve is indicated by that dashed line. And so the difference in oxygen affinity between myoglobin and hemoglobin ensures that the oxygen that's bound to hemoglobin in the lungs is then released to the myoglobin in the muscles. And so this oxygen delivery system is efficient, and that's because the tissue oxygen corresponds to the part of the hemoglobin binding curve where the O2 affinity falls off most sharply. So basically what that's saying is these work together so that we can make sure our different tissues are getting oxygen when they need to. And so hemoglobin actually undergoes conformational changes when it binds to oxygen. And so you can see here that whenever oxygen is bound, the heme group actually kind of pulls up. So whenever oxygen is not bound, it's kind of buried down in. It makes a little bowl. However, whenever it has oxygen attached, it kind of like flexes up. And so kind of what does all this mean? And so oxygen transport, so hemoglobin will pick up oxygen in the lungs, and then it'll go to the tissues, and hydrogen is delivered from the metabolic production of carbon dioxide, and that is going to decrease hemoglobin's affinity for oxygen. So that will then cause and promote oxygen to be released to the tissues. And so then back in the lungs, hemoglobin is able to bind more oxygen and then release the proteins, which will recombine to reform carbon dioxide. And so just kind of thinking, why is this important? So we can think about our a disease that is called sickle cell. And so here you can see normal cells at the center that are rounded, and they can flex whenever they pass through capillaries. But sickle cells, they have an elongated C shape, and they can actually rupture when they're passing through small capillaries. Think about your veins and your capillaries. These are going to rupture. So sickle cell is a disease, and it's a group of inherited red blood cell disorders that affects hemoglobin specifically. And this condition affects more than 100,000 people in the United States and 20 million people worldwide. Normally, red blood cells are disc-shaped, as you can see, and again, they're flexible and can move through. But if you have sickle cell, then these cells are unable to bend and move easily, and they can actually cause your blood flow to be blocked to the rest of your body. And so kind of some symptoms and what happens is that this blocked blood flow through the body can lead to serious problems, including stroke, eye problems, infections, and a lot of people say and have a ton of like chronic pain associated with sickle cell. Unfortunately, sickle cell is a lifelong illness, and right now the only cure is going to be a blood and bone marrow transplant. There are some effective treatments out on the market that can help to reduce symptoms of sickle cell and help to prolong life. And so there's currently a ton of research and a ton of clinical trials going on to help try to find a better cure for sickle cell. So next we can get into structural proteins. All right. And so microfilaments are polymers of actin. And this image is just showing you 3 different types of cytoskeletal filaments. You have actin, intermediate, and microtubules. And you can see they kind of have a different distribution from each other and kind of how they look. And so we can talk about actin. Globular actin, this protein assumes a globular shape, and it has a cleft, which is where ATP will bind. And that those proteins can form actin filaments. And so the structure of F-actin was actually determined by X-ray diffraction and some computer modeling. And so this image is showing you 14 different actin subunits linked together to form a microfilament. And so an actin filament will grow as subunits are added to its ends, and subunits are usually added more rapidly at the plus end, which therefore grows faster than the negative end. And you can see here the original segment is shown in dark, and at the positive you can see that it grows a lot faster than down here at the negative end. And you can also have something called actin filament treadmilling. And so this is when assembly at one end balances the dissociation at the other end. So there's no net change in length, it's just kind of being recycled. And so what happens with this is that as pieces are being dissociated and more pieces are being added, then it kind of appears that these are traveling along the filament like it would be on a treadmill. And so microtubules, these are hollow fibers that are built from tubulin dimers. They alpha beta dimers of tubulin initially form a linear protofilament, and protofilaments will then associate side by side, and then eventually they will form a tube. And so tubulin dimers can add to either end of the microtubule, but growth is again faster on that plus end. And just a note that the hollow tubes usually contain 13 protofilaments. And this is just a really neat picture showing cryo-EM, showing that tubular structure of a microtubule. And so hopefully we've all heard about microtubules. Microtubules can be observed in dividing cells. So here you can see the microtubules in green, aiding in the pulling apart of the chromosomes that you can see in blue. So this is showing an intermediate filament, specifically keratin. And so intermediate filaments are exclusively structural proteins. Again, we can talk about keratin, which is going to be in our hair, nails, and horns. I don't know if you have horns, but my husband says I do. Another one is going to be collagen, and this is one of the most abundant animal proteins. And it has a structural role in holding cells together. So think about the extracellular matrix. You can find collagen in bones and tendons as well. So these are not dynamic. They do not have subunits that can be added or removed. Again, the structure is built and it has remained the same. So it doesn't change. They do twist like or look like twisted ropes. So if we think about keratin, it forms something called a coiled coil, which is a dimer of alpha helices. So you have two alpha helices that will twist together. And it adopts something called a left-handed twist, and it only forms a secondary structure. And that secondary structure, again, those alpha helices, is supported by hydrogen bonds. And so this is just showing an arrangement of the coiled coil. And so here you can see typically there's a pattern of 7 amino acids that are going to repeat and be the structure of the alpha helix. You can also see kind of why it's important here that the first and the fourth amino acids are going to tend to be nonpolar. So when they coil together, the first and the fourth amino acid will be facing inside of the coiled coil helix, and that is going to allow them to interact together. So you can also begin to add these coiled coils together to form tetramers and then form an octamer, and then you start to form the intermediate filaments. So these coiled coils are linked together with disulfide bonds between cysteine residues, and that's what gives strength to the filament. So again, I kind of just talked about collagen, but collagen has a unique amino acid structure. It's 3 helices that twist together. They're considered right-handed triple helix that's formed by 3 left-handed helices. Tropocollagen is what refers to the triple helix. And collagen consists of hydroxyproline, which is a modified proline plus a hydroxyl group. Proline and glycine repeats. So hydroxyproline, proline, and glycine. Glycine is the only amino acid that's small enough to be on the inside of the triple helix. Collagen can also covalently cross-link, and every now and then we will also see a lysine being added in and link together the triple helices. So this is showing you a cross-section of the collagen triple helix. And the thing I want to point out to you here is going to be that the glycine residues, which lack side chains, are going to be marked by the G, and those are what's located at the center of the triple helix. And then the side chains of the other residues are going to point outward from the triple helix. And again, I just mentioned this, so we'll just quickly say it, but collagen is covalently cross-linked, and it does form after polypeptide synthesis. All right, so chapter 8, lipids and membranes. So section 8.1, let's talk about some lipids. So what is the main characteristic that classifies a lipid? So it's going to have a main hydrophobic portion. Again, like we talked about in the first unit, lipids do not form polymers. And so we can talk about fatty acids, which are lipids, and they have long-chained hydrocarbons. And fatty acids are used as a storage molecule, and they can be used as a storage molecule because it doesn't have water. In plants and animals, the even-numbered fatty acids, so for example, C16 and C18 species are going to be the most common. And again, these fatty acids often have long hydrocarbon chains, and fatty acids are considered to be amphipathic or polar and nonpolar. So amphipathic means that you have a polar and a nonpolar group. So the head is going to be a polar carboxylic acid group. So fatty acids, you can also have saturated and non-saturated. So saturated is going to be where there are no carbon double bonds in that hydrocarbon chain. And so saturated fats are going to have a solid characteristic, so they can pack tightly together. They're straight, they can just go in, think about packing up a box of toothpicks. You're going to be able to squeeze a lot in there and make a really solid piece. And saturated fats are going to be solid at room temperature. So next you can have fatty acids that are unsaturated or polyunsaturates. And so double bonds, these are fatty acids that have double bonds that are usually in the cis conformation, and they're going to have at least one double bond that gives a little bit of flexibility in that tail portion. And so these double bonds actually make these fatty acids liquid at room temperature. And so whenever we're naming fatty acids, there are two different ways that you can name a fatty acid. So we have the delta and the omega naming system for fatty acids. So delta, how do we name fatty acids based on the number of carbons and the number of double bonds? So for delta, you're going to start counting carbons at the carboxylic acid end. And so here you can see that we are going to start counting carbons. So we'll start here. So 1, 2, 3, all the way down to 18. And so for delta, this is going to be 18 and then a colon and then 0. So 0, so 18 is the number of carbons and 0 is going to be the number of double bonds. Again, in that top example, we do not have any double bonds, and so that is why we have a 0 instead of a number. All right. And so example number 2, so this is going to be a saturated fatty acid. And so if we're naming it through the delta system, we're again going to count, and I will go ahead and spoiler alert that there are going to be 18. But what I want to point out here is that we have the double bond. And so whenever we're writing it out in delta, it's going to be 18 colon 1. And that's going to indicate that we have one double bond in this fatty acid. And then as a superscript to the 1, you're going to put a delta and then a 9. And so the full name of this in the delta naming system is going to be 18 colon 1 superscript delta 9. And that is just showing that our double bond begins here at the 9th carbon. If you have, for example, 2 double bonds in the delta naming system, then you would have to list where both of those double bonds begin in that superscript. So for example, this one again has 18 carbons, so it's going to be 18 colon 2. You have 2 double bonds, so it'll be 18 colon 2 superscript delta 9 and comma 12. And that's showing that you have 2 double bonds starting at carbon 9 and at carbon 12. I do want to kind of point out that the double bonds are always going to be 3 carbons apart, always. And that's just something to keep in mind. So when we think about omega naming system, you're going to start naming the carbons from the other end, so the methyl group end. And so we can start down here to show how you name with the omega end. Again, there are 18 carbons, 3 double bonds. And so for this, we'll start here. So 1, 2, 3, and that's going to be the start of our double bonds. And that's really all we need to know. Again, because we know that the double bonds are always going to be 3 carbons apart, we can figure it out if we just know where the first one begins. And so for the omega system, this is going to be 18:3. And then you'll also put another 3. And so, or an omega 3. And so 18:3 omega 3. And so this is an omega 3 fatty acid, and its first double bond is at the 3rd carbon. So for the omega naming system, you only have to name where the first double bond is located. So if we look back up at example 3, this name in the omega naming system would be 18:2 omega 6. And that is because the first double bond begins, again, we name it from the opposite direction, but the first double bond begins at carbon 6. All right, so fatty acids are usually found as part of a triacyl glycerol or tag, which can be used for storage energy. So it has an ester bond that links each acyl group, and that's the result of a condensation reaction. So you then have 3 fatty acids on a glycerol backbone that are connected by an ester linkage. This glycerol backbone has 3 hydroxyl groups, and that's where the fatty acids will connect. So if we have 3 fatty acids that are the same, we call those a simple tag or triacyl glycerol. If there are 3 different fatty acids, then we call that a mixed tag. When these proteins are needed for metabolic processes, those fatty acids can then be cleaved off of the glycerol backbone. And this is one of the most nonpolar molecules that we're going to talk about. And so one important note is that it can't be used in the membrane structure due to this. One thing I want to talk about is that some lipids are derivatives of isoprene. And so these can include vitamins A, E, D, and K. But one that is really important is an amphiphilic isoprenoid, and that is cholesterol. So I'm sure you guys have all heard about cholesterol. But cholesterol is important in membranes, and it has a really important ring structure. It has 4 rings, and it's the backbone of estrogen and testosterone. And it's also very highly nonpolar, and it has a small hydroxyl group for that polar group. And so when we talk about a lipid bilayer, these lipid bilayers, again, we just talked about lipids, they're going to be formed spontaneously due to the hydrophobic effect. So remember, your polar head groups are going to want to face out, and your lipid tails are going to want to face in towards each other and be shielded from the water. And so, again, these form spontaneously due to the hydrophobic effect. Proteins, glycerol, phospholipids, and cholesterol are going to be the main components. You have what's called the outer leaflet, which is going to be up here, and that is going to be where you have facing the outside of the cell, and then the inner leaflet is going to be inside of the cell. You can also kind of have, when we think about a simulation of the lipid bilayer, you want things to be able to move around. So you don't want it to be solid. So when we think about lipids and kind of what we talked about, saturated versus unsaturated, whenever you have those double bonds, those can actually add kinks into the lipid chains. And so this is going to be important in adding fluidity to the membrane. And so the double bonds will kink the acyl chain in lipids, And in cold environments, we have more unsaturated or double bonds in the membrane. However, in warm, we actually have more saturated. And this is because saturated fats have a higher melting temperature. And so when we think about melting points of fatty acids, the higher number of carbons with no double bonds or saturated fatty acids, then the higher number of melting point. So for example, 14-0, so 14 carbons with 0 double bonds has a lower melting point than 20-0. So 20 carbons with 0 double bonds. So unsaturated is going to have a lower melting point. So the more double bonds, then the lower the melting point. So one question you may see on an exam would be rank the melting point. So you're going to rank your no double bonds first. So those are going to have the higher melting point. Again, the highest number of carbons is going to be at the very top. And then you can start to add in your double bonds from there. So fluidity is extremely important when discussing membranes. And so when we think about if a phospholipid wants to flip to the other side of the membrane, then it has to go through something called transverse diffusion or flip-flop, which is a very, very slow process. I like to think about this as if you imagine here between the different phospholipids in the bilayer, then this is like a fence. And so it's really hard for these to climb over the fence and flip-flop to the other side. So it's thermodynamically unfavored. Again, it happens very slowly. It can happen, but it's not favored. However, remember, if we think about this as a fence, it's a lot easier to run up and down the same side of the fence than it is to climb over it. And so lateral diffusion is going to be very, very rapid. And phospholipids can go through lateral diffusion where there's movement on the same plane, if you will, quite easily. And so next we'll talk about membrane proteins. So proteins can associate with membranes in different ways. And so remember that within the lipid bilayer, you have lipids, including glyceride, phospholipids, sphingolipids, and cholesterol. You can also have proteins in the bilayer. And so there are different types of membrane proteins, including integral membranes, peripheral, and lipid-linked proteins. And so this schematic is showing you integral proteins that will actually span the width of the membrane. You can have peripheral membrane proteins that are associated with the membrane surface. And then you can have lipid-linked proteins whose attached hydrophobic tail is what incorporates into the lipid bilayer. And so kind of connecting protein structure and function, the portion of the protein that passes through the lipid bilayer must be largely hydrophobic. And so this image is showing you an example of a membrane spanning alpha helix. And so this portion of the sequence is actually from a protein called bacteriorhodopsin. And its 3D structure is showing that the polar residues are in purple and the nonpolar residues are in green. Again, you want the nonpolar residues to be what's going through the membrane. So again, membranes can span the lipid bilayer. So we just talked about an alpha helices example, but you can also have beta sheets or beta barrels that will span the entire membrane. So here you can see that these beta strands, or the secondary structure of the beta strands, are going to have nonpolar amino acids associated with them, and that's what's going to allow them to span together. These beta strands can form almost like like a tube structure that will span the membrane. All right, so fluid mosaic model, kind of like what we're talking about, the membrane is dynamic, and it's always moving. And so integral membrane proteins that you can see, those are going to actually kind of float in a sea of lipids and can move laterally, but again, they can't undergo transverse movement. So just think about these kind of floating around on the sea. They're in boats moving around, and they're going to be constantly moving throughout. However, some membrane proteins may have a fixed orientation. And so proteins can interact with cytoskeleton, and they can be immobile. And so these cytoskeleton structures can actually kind of block in proteins from moving and lock them into place. And so, again, I'm kind of going to go through this part really quickly, but it's important. But, I know you guys have some other great resources as well. This is just a review of it. So, how do ion movements affect membrane potential? So we can use sodium and potassium kind of as a classic example. And so whenever we do this, we need to think about membrane potential. And so membrane potential refers to the difference in charge across the membrane. So you can see that we have a higher concentration of sodium on the outside of the cell here. And we also see a higher amount of potassium inside of the cell. And so if we were to add the charges up, you would find that there is less of a charge inside the cell. And so the membrane potential is going to be negative. So again, remember that the structure of the cell membrane makes it difficult to transport these ions across it because of that membrane hydrophobic core. So we need the proteins, or we need proteins and channels to help for ions to travel through and be moved across the membrane. So we typically see ions moving from a high concentration to a low concentration due to entropy. And the idea is that they're moving to a place with more space. And we call this, so moving from a high concentration to a low concentration, moving down the concentration gradient. There is also a way that you can mathematically quantify the membrane potential, and I, again, highly encourage you to watch the lecture videos to see examples of this. So one example of this is kind of how neurons fire. So you can see membrane potentials changing and causing an electrical signal that can be passed to other neurons. So you can have a stimulus, and that stimulus will then open a sodium channel in the membrane of the neuron. That influx of sodium will cause a depolarization, which triggers the opening of a potassium channel. And so the membrane potential is going to change with these openings of the different channels. And then that initial depolarization is going to trigger the opening of yet another sodium channel, and then again a potassium channel. And so that just continues down the neuron, almost in like a wave pattern. And so the action potential will then move down the length of the axon. And this action potential will only travel in one direction. And so then previously open channels are going to be closed. And so think about it as a pulse through the neuron, but it's just going to be changes in membrane potential all the way down the neuron. And so we can think about passive transport. And so passive transport is going to describe the movement of substances across a cell membrane down their concentration gradients. And the big thing is that it does not require energy. And so structures that can aid in this are porin proteins. These are trimers, and they have pores, hence the name, that allow molecules to pass through. A lot of these porin proteins are mainly beta barrels that span the membrane, and they follow a concentration from high to low. Another example of these are ion channels for passive transport. And a lot of times ion channels are specific for certain ions. Um, Kind of a note on porin proteins. Again, I study the cell biology of a specific parasite. And these can Porin proteins can be important, not only from outside the cell into the cell, but think about, getting into organelles. So the mitochondria actually has a very well-known porin protein, and it helps to get other molecules in and out of the mitochondria. So there's also different types of membrane transport systems. And so you can have a uniport, which is going to be the movement of one molecule independent of other molecules. You can have a symport. And so this is when the movement of two molecules are going in the same direction through a protein channel. And you can also have an antiport. And so an antiport is when you have the movement of two molecules in the opposite direction through a protein channel. And then I'm just going to give you guys kind of the classic example of active transport. Active transport is when you have the movement of ions or molecules across a cell membrane into a region of higher concentration. So you're doing the opposite. So your molecules are moving against the concentration gradient from low to high. One of the classic examples of this is going to be the sodium-potassium pump. And this helps to maintain osmotic equilibrium and membrane potential in cells. So the sodium and potassium move against the concentration gradients. And this sodium-potassium pump will maintain the gradient of a higher concentration of sodium extracellularly and a high level of potassium intracellularly. So it works where the sodium-potassium pump will bind sodium ions. And again, where it's active, it does have to have a form of energy, and that is going to be ATP. And so ATP will quote unquote split and provide energy to change the shape of the channel. So you get an actual conformational change. The sodium ions are then driven through the channel. Once those sodium ions are released to the outside of the membrane, then you have a new shape of a channel. And that new shape is what allows for potassium ions to bind. So once you have the release of the phosphate from that initial ATP, the channel can then revert to its original form. It'll release the potassium ions inside the membrane and the process can begin again. All right. And so with that, it's been about an hour and a half of review. So I am going to end here. Just kind of a good luck from the puppies. Barley, he has been sitting with his head on my lap, listening to me make slides and practice. And then Honey is just staring at me like I'm a big old dingus. So as always, if you guys have any questions, please feel free to reach out. I know this was made kind of last minute, and I deeply, deeply apologize for that. My PhD dissertation is due in 1 week. And so that has been my priority right now, but I am always free if you guys have questions. You can ask your classmates. I've had people send me math problems, and again, I don't have like the technology to write it out, but I will send you pictures of sticky notes where I've written it out. I can be pretty responsive via email. Meeting is a little more difficult, but again, I do have office hours. And once I turn in my dissertation, I will be much more free. And I promise that your next review will be available over spring break. And that way you guys have enough time to listen to it. And then some of you guys, I'll see you for this exam. And then hopefully by exam 3, I will officially have my doctorate. So I'm defending the day before or 2 days before, depending on what class you're in, before your exam 3. So that's really exciting. I've got a lot on my plate, but please do not hesitate to reach out. I also know that your lecturers would love to walk you through some of these. They have posted really great resources and are a great source for you. I highly encourage office hours with them. They're phenomenal people, and you guys are really, really lucky to have teachers and lecturers like them. All right, and so I'm going to end it here, and I wish you guys good luck, and I will see you all as I proctor your exams. So good luck, and I'll see you guys for the exam 3 review in about a month. Bye.